Welcome students, I am Dr. Kim Rodriguez, Assistant Professor, Department of Botany, Kamal College of Arts, Science and Commerce for Women, Nuvengoa. Today I will be teaching concepts, types and practical application of Apomixis for second year BSc Botany Semester 3 students. Course code BOC 103, course title Plant Anatomy and Embryology. Title of the unit, Unit 8, Apomixis and Polyembryony. Module name, Concepts, Types and Practical Application of Apomixis. The module outline is Concepts, Types and Practical Application of Apomixis. The learning outcome of the module is explains the concepts, types and practical application of Apomixis. Concepts of Apomixis. According to Winkler, the term apomixis may be defined as the substitution of the usual sexual reproduction by a form of reproduction which does not involve meiosis and syngamy. A species may include sexually as well as apomictically reproducing individuals. Apomixis is very common in higher plants. Plants of more than 35 families are known to be apomictic. Example, Graminae, Rosaceae, Compositae and Rutaceae. Types of apomixis. There are two main types of apomixis. Vegetative reproduction, agamospermy. Vegetative reproduction. Plants propagate by a part of their body other than the seed. The structural units employed for this purpose are called propagules. Vegetative propagation includes reproduction by means of bulbils, bulbs, runners, suckers, and so on. These propagules are formed by the sporophyte only. Three types of vegetative reproduction have been distinguished in higher plants. Propagules are formed outside the floral regions, and despite the occurrence of functioning sex organs, no fertilization or seed setting takes place. Example, Agave americana and Eloidea canadensis. Propagules are formed outside the floral regions, and the plants are sexually sterile. Fritillaria imperialis and Lilium bulbiferum are typical representatives of this group. They propagate by means of bulbils and bulblets. Propagules are formed on the floral branches, either in addition to the flowers or in place of them. The phenomenon is commonly described as vegetative vivipary. Example, Dyschempsia, Festuca, Poa, and Allium. Agamospermy. Plants have retained seed as the agent of propagation, but the embryo is formed by some process in which normal meiosis and syngamy have been eliminated. Three different types of agamospermy are recognized. Adventive embryony. Embryos arise directly from the diploid sporophytic cells. Sexual embryo sac develops normally, and the zygotic embryo either degenerates or competes with the apomictic embryos. Diplospory. An archisporium differentiates, but the megaspore mother cell develops into an unreduced embryo sac. Embryo is formed by the unfertilized egg or some other cell of the embryo sac. Apospory. A somatic cell in the nucellus directly forms an unreduced embryo sac, and the diploid egg parthenogenetically develops into embryo. Apomictic embryo sac may develop in addition to the haploid embryo sac derived from a functional megaspore mother cell. The latter may either degenerate at an early stage or form a sexual embryo after fertilization. Maheshwari has recognized the following three types of apomixis: non-recurrent apomixis, recurrent apomixis, adventive apomixis. Non-recurrent apomixis. The megaspore mother cell undergoes normal meiotic division, and one of the four megaspores thus formed develops into haploid female gametophyte. However, there is no fertilization, and the embryo develops either from the unfertilized egg or from some other cell like stimulated haploid synergid cell of the embryo sac. The embryo thus formed is naturally haploid. The plants produced by this method are haploid and generally sterile and do not reproduce sexually anymore. Example, Solanum nigrum, Lilium, Nicotiana tabacum. Recurrent apomixis. Embryo sac may arise either from a cell of the archisporium or from some other cell of the nucellus. Nuclei of the embryo sac are usually diploid. Embryo arises either from the egg or from some other cell of the gametophyte. Generative apospory has been observed in Eupatorium glandulosum, Parthenium argentatum. Somatic apospory has been reported in Hyraceum excellens, Hyraceum flagellaire, and Hyraceum aurantiacum. Adventive apomixis. Development of embryo from any diploid cell of the ovule lying outside the embryo sac is referred to as adventive apomixis or embryony. It is also called sporophytic budding. In this process, there is no alternation of generations. The cells of the nucellus or integuments forming adventive embryo become densely protoplasmic and divide actively to form a small mass of meristematic cells. This tissue mass grows actively, pushes its way into the embryo sac, and eventually gives rise to what appears to be a normal embryo. The zygotic as well as the adventive embryo may grow simultaneously in the same embryo sac. The normal zygotic embryo has a suspensor, whereas in the nucellar adventive embryo, suspensor is absent. Citrus is the most common example of adventive embryony. As many as 10 viable embryos may be found in a single seed of citrus. Adventive embryony is also of common occurrence in the members of the Euphobiaceae, Cactaceae, Buxaceae, and Orchidaceae. The development of adventive embryos may take place with or without the stimulus of pollination or fertilization. Practical application of apomixis. Development of pure lines. Apomixis is a means of rapid production of pure lines in plants. Haploid apogamy and parthenogenesis give rise to haploid plants, which after being treated by colchicine treatment can produce diploid pure lines. These pure lines can be used in the production of high-yielding cultivars and hybrids. Maintenance of purity. Obligate apomixis breeds retain the characteristics of the mother plant, which are useful in maintaining genetic purity from generation to generation. It can maintain a genotype for several generations. Conservation of heterosis. Obligate recurrent apomixis is useful in conserving hybrid vigor for numerous generations. As apomixis does not permit segregation, heterosis can be easily conserved. Easy hybrid seed production. Apomixis offers a simple way of hybrid seed production. These seeds are automatically produced by apomictic means, and there is no need of crossing. Once a hybrid is developed using an apomictic line as one of the parents, the hybrid seed production will occur automatically. It is also more affordable than conventional methods of hybrid seed production. To summarize, apomixis may be defined as the substitution of the usual sexual reproduction by a form of reproduction which does not involve meiosis and syngamy. As apomixis does not involve meiosis, there is no segregation and recombination of chromosomes. Thus, it is useful in preserving desirable characters for indefinite periods. In obligate apomictic species, though desirable characters are preserved for quite a long time, they are deprived of evolution. On the contrary, in facultative apomictic species, sexual and asexual processes occur simultaneously, and hence there is a great significance of apomixis. These are some of the references. Thank you.